Two enormous snakes can look so similar that even experts cannot reliably tell them apart. Same olive green body, same black oval blotches, same eyes and nostrils riding high on the head, same habit of disappearing into dark tropical water. But inside their cells, one part of their DNA tells two very different stories. In 2024, researchers proposed that the green anaconda was not one species spread across northern and central South America. They argued that it was two. The northern lineage was named Eunectes akayima, the northern green anaconda. And the strange part was not that scientists had found an unfamiliar-looking monster. It was that this supposedly new species had been hiding in plain sight. These snakes were already known. People had encountered them, photographed them, studied them, and sometimes captured them. The surprise came when researchers compared genetic samples collected across South America. Snakes that appeared almost interchangeable separated into two major branches on a mitochondrial family tree. This is the mystery of a cryptic species. A cryptic species is not necessarily rare, tiny, or recently evolved. It is a lineage that looks so much like another lineage that appearance alone can conceal the difference. Imagine placing two green anacondas side by side in shallow water. Their patterns vary naturally. Their proportions change with age and sex. Mud, reflections, and vegetation hide finer details. Even scale counts can overlap. To human eyes, they can look like members of the same species. DNA can preserve a split that skin and scales do not clearly display. The 2024 study combined new samples with existing genetic material from anacondas across 9 countries. Some of the most discussed samples came from the Bameno region of the Baihuayeri-Waurani territory in Ecuador. Waurani co-authors Marcelo Tepenya Baihua and Pentti Baihua were credited with helping capture anacondas there. That collaboration mattered for a practical reason. A map may show a river as a thin blue line surrounded by green. On the ground, that line becomes a maze of flooded forest, hidden channels, tangled roots, and water with almost no visibility. An anaconda can remain motionless with nearly its entire body submerged. Finding one requires far more than arriving with laboratory equipment. It requires knowledge of the river and the animal's landscape. The researchers focused on 3 mitochondrial genes called cytochrome B, ND2, and ND4. Mitochondria are the energy-producing structures inside cells, and they carry a small genome of their own. In most animals, mitochondrial DNA is inherited through the maternal line. That makes it extremely useful for tracing ancestry. Mutations slowly accumulate as populations become separated. Compare enough mitochondrial sequences, and clusters begin to appear. Think of it as following one ancient thread through generations of mothers. If two populations have not shared that thread for a very long time, their mitochondrial sequences can become noticeably different. The northern and southern green anaconda lineages differed by about 5.5% across the mitochondrial sequences analyzed. On the researchers' tree, the samples did not form one mixed cloud. They formed 2 strongly supported sister clades. One northern group included samples from Ecuador, Venezuela, Trinidad, Guyana, Suriname, and French Guiana, with the researchers also expecting it in Colombia. The southern group included samples from Peru, Bolivia, Brazil, and French Guiana. French Guiana appearing in both groups is important. It suggests that the real boundary may not be a clean horizontal line across a map. The lineages could approach each other, overlap, interbreed, or meet in a narrow contact zone. Without dense sampling from those regions, the map remains incomplete. The researchers proposed the name Eunectes akayima for the northern lineage. The word akayima is connected to Caravan words for a great or large snake. The familiar name Eunectes murinus was retained for the proposed southern species. But why would 2 lineages remain visually similar after spending so long apart? Because separation does not automatically force animals to look different. Both lineages occupy warm wetlands, flooded forest, marshes, slow rivers, and vegetated shallows. Both face the same basic problem. A huge snake must approach prey without advertising that several meters of muscle are moving through the water. The green body breaks against aquatic plants. Dark blotches interrupt the outline. Eyes and nostrils on top of the head allow the snake to watch and breathe while keeping most of its body submerged. A thick, powerful body works in water, where buoyancy supports mass that would be harder to move on land. If that design continues to work in both regions, natural selection does not need to repaint either snake. Their genes can drift apart while their external appearance remains under similar pressure. The 2024 researchers also estimated that the northern and southern mitochondrial lineages separated millions of years ago, possibly during the Miocene. They proposed that ancient changes to northern South America's rivers and wetlands helped divide ancestral populations. The Amazon and Orinoco systems were not always arranged as they are today. Rising land, shifting drainage, enormous wetlands, and changing connections between basins repeatedly divided and reconnected aquatic habitats. For an animal tied to shallow water, a geological change does not need to create a towering mountain wall. Redirecting a river or draining a wetland can split a population just as effectively. Over generations, snakes on opposite sides accumulate different mutations. Eventually, their mitochondrial histories appear as separate branches. That is the powerful part of the northern anaconda proposal. It reveals a deep geographic pattern inside an animal that seems capable of moving through one gigantic connected wetland. But a separate mitochondrial branch is not automatically a separate species. Mitochondrial DNA follows only the maternal line. It does not represent the entire genome stored in the cell nucleus. Two populations can carry old mitochondrial lineages while still exchanging nuclear genes. Past hybridization can move mitochondrial DNA from one population into another. Large ancestral populations can also preserve old genetic branches long after other parts of the genome have mixed. So mitochondrial DNA is excellent at revealing a mystery. It does not always solve that mystery by itself. Soon after the 2024 announcement, other taxonomists challenged both the evidence for the split and the way the new name had been established. Critics argued that the original study needed stronger tests using informative nuclear DNA, broader sampling near possible contact zones, and a more rigorous comparison of physical traits. They also pointed out that 5.5% mitochondrial divergence has no universal meaning. In some snakes, recognized species differ by less. In other cases, populations treated as one species differ by more. There is no genetic odometer that reaches a fixed number and flashes new species. A species is an evolutionary lineage, not a percentage. Researchers want to know whether the two groups remain independently evolving where they meet. Are they exchanging genes? Do they choose different mates? Are there consistent anatomical, ecological, or behavioral differences? Or are they one widespread species carrying two old maternal histories? Then there was a second argument about naming. Zoological names follow formal rules involving old descriptions, type specimens, publication requirements, and priority. Green anacondas have accumulated several names over centuries. Critics argued that older names and historical specimens complicated whether akayima was the correct available name, even if the northern lineage eventually proved to be a separate species. The original researchers responded later in 2024. They published a revised description, defended Eunectes akayima, and presented proposed differences involving body dimensions and tail proportions. They also listed molecular characters that distinguished the mitochondrial lineages. The debate did not vanish. By 2026, the split was still not universally accepted, and the reptile database treated Eunectes akayima as a preliminary synonym of Eunectes murinus. That does not mean the DNA pattern disappeared. The northern and southern mitochondrial clades are real results in the sampled data. What remains disputed is the conclusion attached to them. Do those branches mark two species, strongly structured populations within one species, or a more complicated history involving contact and gene flow? Answering that will require more snakes, especially from the spaces between the known samples. Researchers need nuclear genomic data from both lineages and from suspected contact zones. They need physical measurements linked directly to genetically identified individuals. They need to discover whether the two forms overlap, and if they do, what happens when they meet? The conservation consequences could be serious. One enormous green anaconda species spread across much of tropical South America has a huge apparent range, divided into separate lineages, and each inherits a smaller range, different threats, and possibly a different conservation outlook. Protecting the wrong map can hide local declines. A population may look secure because similar snakes survive thousands of kilometers away, even if those distant animals belong to a separately evolving lineage. And that is the final reversal. DNA did not reveal a bizarre new snake with horns, stripes, or a never-before-seen hunting weapon. It revealed that the familiar green anaconda may be stranger than that. Its body can cross rivers. Its lineage may have been shaped by rivers that vanished millions of years ago. The northern green anaconda is therefore more than a new name. It is a live scientific question, wrapped in olive scales. The snake has not changed. What changed is our ability to see the history hidden inside it. From the surface, there is one giant animal in dark Amazon water. Follow the maternal DNA beneath that surface, and the snake splits in two.